Welcome you all in CEC-UGC Gurukul live lecture. Learners, we are discussing here biopolymers, especially the chemistry of nucleic acid, chemistry of DNA and chemistry of RNA. And in this part, we will discuss purines and pyrimidine nucleotides, biosynthesis and metabolism of pyrimidine and purine nucleotide, degradation or metabolism of purine, degradation of pyrimidine, ATP, ADP and AMP. What are these molecules? Adenosine triphosphate, adenosine diphosphate, which is ADP, and adenosine monophosphate, which types of nucleic acids they are. So, as I told you, there are two types of bases present in the nucleic acids, that is, one is purine basis and pyrimidine basis. Along with that basis, some minor purines and pyrimidine bases also present in a very small amount, and these are methyl derivatives. So, on 6 carbon nitrogen atom there is some methyl groups. So, these methyl purines, derivatives of purines and pyrimidines are present in small amounts. Now, both the bases, purines and pyrimidine bases, are not related metabolically. Distinct pathways for the purine biosynthesis and degradation of purine, also biosynthesis of pyrimidine and degradation of pyrimidine exist in all living organisms. So, as I told you, there are 3 pyrimidine bases. One is cytosine, thymine and uracil. Also, there is a 2 purine bases, adenine, guanine, and these are the 2 bases. Now, we will say here biosynthesis of purine base. How purine bases can be biosynthesized. So, if we talk about the any nucleotide or nucleosides, we have to consider sugar plus base, because the nucleosides having sugar plus base. So, learners, the purine biosynthesis, de novo synthesis of purine is the most active in liver, which export the bases and nucleotide to the other tissues, and it is a multi-step process. Almost 10 steps takes to biosynthesis of the purine base. So, first it starts from the ribose sugar, but learners, it is very important to understand here, the ribose sugar in its original form, it is inactive. So, here we need a ribose 5-phosphate. So, purine ring is built up step by step by ribose 5-phosphate, which is used as a primer. So, the ribose 5-phosphate on oxidation of pentose phosphate pathway, it is needed for the synthesis of nucleotide and nucleic acid, which is DNA and RNA. So, this is the structure of ribose 5-phosphate. It is a pentose sugar, and at 5 carbon atom you can see the H is replaced by 1 phosphate group. So, it is ribose 5-phosphate, which is a primer structure, or we can, the reaction of biosynthesis start from this sugar, ribose 5-phosphate. Now, the enzyme we required here is 5-phosphoribosyl 1-pyrophosphate synthetase. So, this is an enzyme, we called it PRDF. So, in your screen you can see. Now, what happened? Here the anomeric carbon of that sugar transfer of pyrophosphate group from the ATP to C1 of ribose phosphate sugar. So, on your screen you can see the structure. At C1 carbon atom there is these 2 phosphate group transfer from the ATP molecules. Now, next enzyme is PRPP amidotransferase. So, phosphoribosylamine replaces the phosphate group which is present at the C1 carbon atom. So, that enzyme replaces the phosphate group with nitrogen from the site chain of the glutamine, and this reaction is committed step for the purine biosynthesis. So, here it is very important understand we required here first we required here ribose 5-phosphate, and along with the ribose 5-phosphate we required here some amino acids like glycine, aspartic acid and glutamic acid, which is used for the biosynthesis of adenine and guanine. So, purine ring is constructed from simple building block, that is THF, tetrahydrofolate. So, it is the first nucleotide formed in the pathway, which is the IMP, inosine monophosphate. So, learner, inosine monophosphate, it is the precursor of purine biosynthesis, and the level of this IMP is controlled by the guanine adenine. So, these bases AMP and GMP, these are present in the equilibrium. Another step in the biosynthesis is adenyl succinate, which is converted into inosine monophosphate, and inosine monophosphate is a nucleotide which play important role in purine metabolism. It is also called inosinic acid, and it is used to store chemical energy in muscles and other tissues. So, these are the as I told you, the purine biosynthesis is a multi-step process. So, as we had seen in the previous slide that IMP is converted to xanthosine in presence of that enzyme convert into XMP. This is xanthosine monophosphate, and here we need a certain amino acid. In presence of that, the level is controlled, IMP is controlled by that amino acid, and we get a guanosine monophosphate, that is a GMP. So, in ribonucleoside, though in here in ribonucleoside means which are having a ribose sugar. So, ribonucleosides monophosphate formed by the purine or pyrimidine metabolism to ribose and a phosphate group. So, on your screen you can see here, this is a base, purine base, and that base having a ribose sugar. That ribose sugar is on phosphorylation gives the ribose pyrophosphate. So, how the purine metabolized in living organism, or we call this degradation of purine. Learners, all the purines, most of the purines in human body degraded into uric acid. So, the degradation of purine through the hydrolytic removal of phosphate group and nucleotides cleaved into bases and ribose 1-phosphate, and that nucleotides after cleaving convert into uric acid. So, purines converted into or metabolized into or degraded into uric acid. So, the uric acid is the end product in human. Uric acid is synthesized by xanthin oxidase via hypoxanthin and xanthin. This enzyme is FAD, contain FAD, non-heme iron and molybdenum. So, on your screen you can see how the purine metabolized into uric acid. So, this is the scheme. In this scheme we can see the steps how adenosine monophosphate convert into uric enzyme, and the steps, it is also multi-step process. Inosine ribose 5-phosphate convert into hypoxanthin, hypoxanthin presence of oxidase, xanthin oxidase enzymes convert into xanthine, and finally uric acid. So, purine is metabolized to uric acid in human biological system. Learners, between 500 and 800 milligram of uric acid are formed in human body per day, and most of it in the liver. Intestine also produced uric acid mainly from dietary purine. So, the purine which we take from the diet, that intestine produce that uric acid from the dietary product. Now, about 70% of uric acid extracted by our kidneys, while 30% of uric acid extracted by intestine, and it is metabolized to the other products by intestinal bacteria, because our intestine having a certain type of bacteria. So, 30% of excreted uric acid which is excreted by the intestine metabolized to other products by the intestinal bacteria. Instead of being degraded to uric acid, free bases can be recycled into nucleotide pools, and this requires enzymes, that is hypoxanthine guanine phosphoribosyl transferase. So, this is an enzyme which is called HPRT and adenine phosphoribosyl transferase. So, APRT. So, these are the enzyme which degraded uric acid into free base and can be recycled into nucleotide pool. Learners, about 90% of purine bases that form during the catabolism process of nucleotide are not degraded to uric acid but recycled to nucleoside monophosphate, and this is the here biosynthesis of pyrimidine base. So, in this slide we are looking here how the pyrimidine bases are biosynthesized, and the pathway states with the carbamyl phosphate and aspartate ortic reactions. So, here different domains of single large polynucleotides forms. Now, just like told you the purines degraded to uric acid, similarly pyrimidine bases also metabolized or degraded into water. So, it is degraded to water soluble products that are either excreted as such or oxidized to carbon dioxide and water. So, the end product of degradation of pyrimidine is water soluble products or carbon dioxide or water. So, what is the structure of nucleosides? Learners, nucleosides in 1909 by Levine and Jacob to denote the carbohydrates derivatives of purine bases isolated from the yeast nucleic acid by hydrolysase enzyme. Later, for term was expanded to additionally cover compounds pyrimidine and purine bases, but today it is applied to all the naturally occurring and synthetic compounds, essentially N and C glycosides derivatives of various carbohydrates and heterocyclic compounds. So, nucleosides are glycosylated purine or pyrimidine base. So, on your screen you can see here it is a glycosides. Clearly you can see it is a pentose sugar and anomeric carbon, that is beta carbon, is attached. It is having base, and there are two types of that pentose derivatives, that is one is ribonucleoside, other is DNA, deoxyribose nucleosides. So, when number 2 carbon, one carbon anomeric carbon, and next to that carbon you can see on your screen, when this OH is replaced by H, we called deoxyribose sugar. So, here 2 deoxyribose, you can see here in the structure, the OH is replaced by H, anomeric carbon. On that anomeric carbon, base is joined through the glycosidic linkage, and at C2 position there is a H in case of deoxyribose sugar. deoxyribonucleosides, and the N9 of purines or N1 of pyrimidine bases are linked to beta anomeric carbon. So, you just remember where the bases are bonded on to the ribose sugar. Ribose sugar can be deoxyribose or ribose, just depending upon the number 2 position of carbon atom, whether it is having H or OH. So, on your screen you are looking here, beta anomeric carbon is having a purine. So, it is a purine nucleoside. So, difference is that if X purine basis, as I told you in the previous slides, the purines are double cyclic structure, and the difference is that if X having OH or NH2 or Y is having H, OH or NH2, while in pyrimidine nucleoside that is also having a anomeric carbon atom. So, on anomeric carbon atom there is a glycosidic bond. On that glycosidic bond can be pyrimidine or purine bases, and the sugar can be ribose sugar or deoxyribose sugar. This is the structure of nucleosides. The nucleosides can exist as a syn configuration. So, here syn configuration of nucleosides exists in two conformations or two configuration on the account of constraint of steric situation of bases and sugar around each other. So, there can be syn or anti-pentose of nucleosides with a prime adenosine. It is the structure of guanosine, it is a guanosine base plus ribose sugar. Here we can see the ribose sugar or deoxyribose sugar is having thymine or thymidine, cytosine or cytidine, uracil or uridine, which is a syn configuration. Without hindrance, the bases can rotate about the glycosidic bonds. So, that is the glycosidic bond is between beta anomeric carbon of the sugar and nitrogen of the base. So, in the syn configuration, because there is no hindrance, so the base can rotate towards the glycosidic linkage around the glycosidic bond. Similarly, on your screen you can see the anti-conformation are preferred over the syn for base forming two strand of DNA. So, in the DNA molecules in double helical structure, anti-conformation of nucleosides is preferred over the syn. Ribosides or ribonucleosides, syn conformations are found in coenzyme syn adenosine, syn guanosine, syn cytidine, syn uridine and syn thymidine. Similarly, in ribonucleoside, anti-conformation found in RNA, anti-adenosine, anti-guanosine, anti-cytidine or anti-uridines. In case of deoxyribonucleosides, anti-conformation found in DNA, it is called anti-deoxyadenosine, these are the nucleosides, anti-deoxyguanosine, anti-deoxycytidine and anti-thymidine. So, these are the bases, adenosine, guanosine, cytidine and thymidine, these are the bases, and when they present in an anti-conformation, we call it as anti-deoxyadenosine, guanosine, cytidine and thymidine. Now, structure of nucleotides. As we had discussed in the previous slides, in case of nucleosides, you just see the difference. In nucleoside, we have seen the C5 carbon atom having a OH group, but learners, the nucleotides are phosphopyrolylated nucleoside. It means that the sugar can be ribose sugar or deoxyribose sugar, but the phosphate group present at C5 carbon atom. So, that is replaced by C5 carbon atom. H is replaced by, there is a CH2OH, but H of CH2 group is replaced by phosphate group. So, and the same as in nucleosides, the base are present on the anomeric carbon atom. So, this is the difference between nucleosides and nucleotides. So, or we can say nucleotides are phosphopyrolylated nucleoside sugar. DNA bases are having same bases are present can be here adenosine, cytosine, guanine and thymine. In RNA, uracil, cytosine, guanine and thymine. So, you can see the difference of bases here, only uracil. In DNA there is a thymine, and in RNA there is a uracil, while DNA having thymine. DNA and RNA, both the molecules are polymers and made up of a long chains of nucleosides. So, this is the structure of a nucleotide. On your screen you can see the structure of nucleotides. So, it is very easy to draw. How you will draw? So, you just at anomeric carbon atom you draw the base, any base, that can be pyrimidine base or that can be purine bases. On your screen you can see here the sugar is a pentose sugar, and that sugar can be ribose and deoxyribose. At C5 carbon atom there is a phosphate group, and at anomeric carbon atom there is a base. So, base you are looking, nitrogenous base you are looking here, thymine. So, phosphate, if there is a one phosphate group, it is called monophosphate. When there is a 2, it is diphosphate. When there is a 3, it is a triphosphate. And there is a ester bond. You can see on CH2, O and P. So, between O phosphate group there is a ester bond. So, ester linkage is having 2 for phosphorylation of nucleotide or nucleosides. So, nucleosides is nucleogenous base plus sugar. And how you have to represent the nucleotides? We can represent nucleoside plus phosphate. So, just difference is a phosphate group at C5 carbon atom. So, nucleotides are also named as nucleoside mono, di or triphosphate on the basis of phosphate group. Example, adenosine monophosphate AMP, adenosine diphosphate ADP or ATP, adenosine triphosphate. So, these are the nucleotides which are having 1 phosphate group, 2 phosphate group and 3 phosphate group. So, ATP is a adenosine triphosphate which is having 3 phosphate group. So, this is the structure of a nucleotide. So, you can easily see here ATP, ADP and AMP. So, ATP is having all the 3 phosphate group, ADP having 2 phosphate group and AMP having only 1 phosphate group. So, sugar plus base plus 1 phosphate group, 2 phosphate group and 3 phosphate group. So, ATP is the energy currency of cell, and it is a metabolic process is derived from, all the metabolic process is derived from the ATP. It is a act as coenzyme precursor of DNA and RNA synthesis. NAD, nicotinamide adenine dinucleotide or NADP has an essential roles in redox reaction and act as an electron donor carrier. NADP involved in anabolic process. ATP is a carrier of chemical energy. Learners, on your screen you are looking here the structure of ATP. It is also called energy currency. So, adenosine 5 triphosphate is known as universal carrier of chemical energy because the energy of hydrolysis of ATP converts all the endergonic reactions into exergonic reactions. So, energy is a glucose and lipids converted into ATP. Importance of ATP to biological reaction is shown by its turnover rate in humans. Each day a person uses as an amount of ATP equivalent to his or her body weight. So, on the basis of our body weight, we required amount of energy or ATP. Enormous amount of ATP is continually being broken down and synthesized in biological organisms. And energy is released. How the ATP release energy? Learners, because ATP is having a 3-phosphate group. So, when the third phosphate group is broken, energy is released because the third phosphate group is unstable and the bond linking to phosphate group have high potential energy. So, the third phosphate group in ATP is hardest to keep bonding and it's needed a most of energy. So, it quickly broken down and release a lot of energy. It is a glucose ATP cycle. In ATP cycle, ATP are needed every second by a cell. So, ATP is created inside them due to demand. So, glucose or sugars delivered via blood streams, which is a product of food, used to create ATP, and glucose is broken down in a series of enzymes which is controlled steps, and that steps allow the release of energy used by organism, especially respiration. So, because our respiration also required energy, during our respiration we need energy. So, this is the process, this is the ATP cycle. We can use the energy during the ATP cycles. So, there are certain types of nucleophilic substitution reaction takes place, and the nucleophilic attacks breaks the phosphate anhydride and rather than pi bond, because the phosphoanhydride easily broken down. So, this bond is easily broken down due to nucleophilic attack. So, ATP plus D-glucose phosphate, and here the OH group of glucose attack the terminal phosphate of ATP and it's break the phosphoanhydride bond without forming an intermediate. So, it is a inline displacement mechanism. So, it is a breaking of phosphate anhydride bond. On your screen you are looking the reactions where the breaking of phosphate anhydride bonds takes place, and it is a very important question, why phosphorylation of glucose requires ATP? Because without ATP the OH group of glucose would have to displace a very basic OH group from the hydrogen phosphate. So, with ATP, OH of D-glucose displace the weakly basic ADP. So, it is the main reason phosphorylation of glucose requires ATP. So, the transfer of phosphate group from ATP to D-glucose, phosphoryl transfer reaction takes place. So, the overall reaction is there are two reactions, one is endergonic reaction and other is exergonic reaction. So, endergonic reaction is a positive while the exergonic reaction is a negative, but the overall final reaction, net reaction, is a exergonic. So, energy is released from the hydrolysis of ATP is more than enough to derive the phosphorylation of D-glucose couple reactions. So, learners, in this part we had discussed about the structure of nucleoside, structure of nucleotides and structure of ADP, AMP and ATP. Why ATP is why ATP is unstable, and we have learned from the various reactions how the reaction takes place, how the phosphoanhydride bond broken down during the hydrolysis and release a lot of energy. Thank you.